Today we'll be looking at the process of transcription in a prokaryote. Transcription is the process of a DNA strand being copied into a new molecule of mRNA, which is messenger RNA. Transcription is the process of how we get DNA to RNA. Here we have a DNA molecule. Our blue strand is our coding strand, and our tan is our template strand. The coding strand is a non-template DNA strand that has the codons that will be used to determine what the protein is. The exact sequence, but the T's are replaced with U's. Template strand has to be anti-parallel and is read 3 to 5. Transcription is broken down into 3 steps: initiation, elongation, and termination. Keep in mind, during this whole process, new nucleotides are always added at the 3' end. RNA is built from 5 to 3 and read 3 to 5. So here's a DNA strand wanting to be transcribed. First, we will have our RNA polymerase with the sigma factor. This is called the holoenzyme because everything is included. Sigma factor bonds onto this region and signals for the RNA polymerase to bond to the promoter region of the DNA to begin to separate the strands in initiation. The Tata box is 10 upstream and the control 35 upstream. This is just the core enzyme. RNA polymerase is composed of protein subunits. When it interacts with the sigma factor, it is called the RNA polymerase holoenzyme. Once separation happens, elongation is started. Step 2. RNA polymerase moves down the template strand and reads each base. The new mRNA is the same as the coding strand. Thymine is replaced with uracil. As we can see here, the T's are replaced with the U's in mRNA, and these two, everything is complementary to each other. The promoter is located on the 5' end and has the 35 region and the 10 region, also known as the Tata box, and they have the instructions for RNA polymerase. These regions are what is recognized by that sigma unit. Okay, now let's keep going. DNA is unwound at the front of the bubble and rewinds at the back. As the RNA polymerase moves down the DNA strand, DNA polymerase adds ribonucleotide triphosphate from 3' end until all are transcribed. Phosphodiester bonds between ribonucleotide triphosphates are stemmed. RNA is transcribed from the 3' to the 5' end, and again, the nucleotides are added from the 3' region. Termination hairpin is made and because of an influx of adenines are at the end, and an AU pair is made, which are weak and can quickly terminate. mRNA is then dissociated and goes to the ribosomes to make strong proteins. If there was no Tata box, no new bases would be able to access the template strand. In the 10 region, thymine and adenine are composed. Without it, there would be a very low level of transcription. RNA polymerase would bind, but not much would happen, and much of what would be transcribed would die off. So this is what it would look like again. Also wanted to make a note that the +1 region, which was 1 downstream, is the first base that is used as a template to make the first base in the RNA.